In this video, we will discuss about one of the disease that is related to muscle, that is human skeletal muscular hypotonia. So this is related to weak muscle that is sometimes also called as sponginess. So let us first understand the normal muscle function and the protein that is essentially plays a essentially or plays an important role is stim1. So how this stim1 perform a function to develop a good muscles is by maintaining the calcium levels or calcium homeostasis. So there are different ways by which stim1 can maintain the calcium homeostasis. Stim1 is a membrane protein that is found on the membrane of endoplasmic reticulum, as it is shown here. This stim1, and it interact with oral 1 and DHPR, and these oral 1 and DHPR are found on cell membrane. So what happens is that when stim1 interact with oral 1, then it will initiate the calcium entry into the cytosol with the help of SOCE, that is known as storage operating calcium entry. So this oral one, when interact with the stim1, it promotes this entry of calcium from T tubules membranes to the cytosols of the cells. And simultaneously, stim1 also interact with DHPR during EC, that is extracellular you can say external contraction. I'm not able to recall the exact full form, but it is related to contraction, extracellular contraction, maybe like this. So this contraction leads to interaction of DHPR and RYR1, and this interaction will helps in the transport of calcium from sarcoplasmic or we can say endoplasmic reticulum to the cytosols, and and to control and to or attenuate this Ca2 flow, the stim1 will act as a signal that attenuate or controls the calcium homeostasis from this SR sarcoplasmic or endoplasmic reticulum to calcium homeostasis. And in this way, in regular normal condition, the uh, stim1 act as a essential uh, we can say and membrane protein on endoplasmic reticulum to control the calcium homeostasis. So however, what happens in the mutated form of stim1 and two and one mutated the mutated form that has been studied is replacing R at 429 amino acid position of stim1 from N terminal to C. That is, arginine is replaced with cysteine at 429 position of stim1 protein from N terminal. So what happens in this case is that the interaction of the stim1 with ORI1 and oral 1 and DHPR cannot be established. Because of this, what happens is that the normal function in which Ca2 transport from oral 1 into the cytoplasm by interaction with stim1 will get abolished, and there is no Ca2 plus uh, there is no Ca2 plus inflow within the cytosols. However, in other case in which during the contraction the DHPR interact with RYR1, and this triggers the Ca2 plus transfer from endoplasmic reticulum or sarcoplasmic to cytosols. However, since there is no attenuating signal available, as stim1 is not interacting with DHPR in mutated form, so the higher flow rate of Ca2 plus or calcium will flow into the cytoplasm, and this will create a low Ca2 plus condition in the uh, sarcoplasm or cytoplasm, and this condition will lead to several problems, such as one of the problem is abnormal mitochondria. uh, Abnormal mitochondria functioning has been observed because of high Ca2 plus in the cytosols, and because of this the ATP formation of mitochondria will also be reduced, and we know from our childhood, from our school classes, that mitochondria is known as powerhouse of the cells, and ATP is and it generates energy in the form of ATP. And when this get affected, this will cause uh, energy deficient or energy disproportion within the cells, or we can say the energy required for normal functioning of the cells will not be supplied by the mitochondria. And other problems because of low Ca2 plus in endoplasmic reticulum is also initiate ER stress that will cause a unfolded protein response that leads to epigenetic changes. Uh, so that will cause another cascade problems on the muscles and all on the cells. So these are one of the also this is uh, one of the problem of there is this is one of the reasoning, we can say, understanding of how this hypotonia works, and the importance of stim 1 protein to maintain the normal muscle functioning. I hope you have understood something about this human skeletal muscular hypotonia. And if you need to understand more about this problem in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.